So, welcome everybody to the CADASIL Connection webinar series. On behalf of myself, I'm Bert Kosiski, the president of cureCADASIL, and I have Anita Ball today, who is a volunteer who's been helping us set up these webinars. And I'll remind everyone that, or tell everyone that you are muted during this presentation, but we're hoping that you have questions along the way, and you'll type your questions in the drop-down question box. And at the end of the session, we'll have a question and answer period in which we'll go over those questions. So cureCADASIL is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients in the medical community toward the common goal of treatments and ultimately a cure for this rare genetic disease by promoting communication, advocacy, research, and education. So we're fortunate to have our speaker today, Dr. Marc Vila Cuenca, who is a senior postdoctoral researcher at the Orlova Lab and CADASIL research group at the Leiden University Medical Center in the Netherlands. Dr. Cuenca received his PhD studying vessel dysfunction and chronic kidney disease at Amsterdam University Medical Center, where he developed a strong interest in vascular disease and disease models. In his current position, he works with induced pluripotent stem cells-based 3D vessel-on-chip system to model heterocellular endothelial and vascular smooth muscle cell crosstalk during development and disease. So today, CADASIL, he's going to speak on CADASIL being the most common form of hereditary stroke disease caused by mutation in the Notch3 protein. Currently, CADASIL laboratory experiments are based on genetic mice models or simple human cell cultures which do not replicate the complex structure of the human blood vessel. The results from these laboratory models are difficult to translate in the clinical setting. Vessel-on-chip is an emerging technology containing a miniature vessel which allows our understanding of human blood vessel during development and disease. By generating vessel-on-chip containing CADASIL patient cells, they will provide an alternative experimental tool to understand CADASIL disease. With this model, they can see how Notch3 mutations affect the structure and functionality of the miniature vessels. In addition, they show that this model can be a useful platform to study therapeutic approaches in CADASIL disease. Therefore, this vessel-on-chip CADASIL model captures patient blood vessel physiology in a more relevant manner, which allows a better understanding of CADASIL disease and helps to develop predictive methods to investigate drug disposition. So, without further ado, I'll try to switch this over to Marc. Great.
Thank you, Bert. I hope that you can see my screen.
Yes, it looks good.
Wonderful. Good afternoon to everybody. If you are in Europe, good morning. First of all, thank you very much for the cureCADASIL organization for allowing me to present some data here in this webinar series. For this presentation, I would like to talk about how we are generating a CADASIL vessel-on-chip model, which incorporates induced pluripotent stem cells, derived vascular cells coming from patients. And we aim to use this model not only to study and understand CADASIL disease, but also in the future to use it as a platform for drug development as well. So for this presentation, I would like to start with the basics. First of all, describing the human blood vessel architecture, the composition of endothelial cells and smooth muscle cells, why this interaction is important, how Notch3 plays a role in this, Also because it's relevant to CADASIL disease, and overall, why do we want to use vessel-on-a-chip technology in order to recapitulate all this human blood vessel structure? So we know that the human blood vessels is composed mainly by endothelial cells and vascular smooth muscle cells. Endothelial cells form the inner lining of the blood vessels, and the smooth muscle cells around this inner lining of the endothelial cells, allowing the cell-to-cell interaction, which is important not only for vascular development and therefore the recruitment of the cells and formation of the vessel, but also for the maturation, which means that it will be a remodeling. Also, endothelial cells and smooth muscle cells will change phenotype. Smooth muscle cells will modulate towards more contractile acquisition of characteristics, and this is important for regulation of the vessel function. So there are several pathways that regulates these changes in the vessel, but one of them, Notch3, it is important and it is also relevant for CADASIL disease. Notch3 is a transmembrane protein that is predominantly expressed in vascular muscle cells, and it will be recognized by the JAK delta family ligands that can be expressed by the endothelial cells. However, Notch3 also can be activated by adjacent smooth muscle cells. Upon interaction with the ligand, this will trigger the cleavage by gamma secretase of the extracellular Notch3, which is suggested to be released in the extracellular, the external part of the smooth muscle cells, while the intracellular domain will be translocated into the nuclei, and it will regulate differentiation and maturation of the vascular smooth muscle cells through gene expression. Among the genes that can be upregulated after the action of the cellular domain of Notch3, we have the K and Hs family, but also as a positive feedback loop, we have more expression of Notch3 protein and also proteins that are related to the maturation of smooth muscle cells and phenotype, like the PhD receptor beta, which is important for proliferation of vascular smooth muscle cells and also contractile markers. And therefore, this will trigger the activation of contractile genes and the acquisition of a contractile phenotype in smooth muscle cells. In CADASIL, however, mutations are placed in the extracellular region of the Notch3, and in particular, mutations are known to affect the secondary structure of the epithelial growth factor like repeats. There are 34 in total in this extracellular domain. And in a regular situation, we do have a fair number of cysteines. However, CADASIL mutations lead to an addition or deletion of cysteines, which will affect the secondary structure of this EGFR domain, and eventually will trigger the recruitment of other Notch3 extracellular domains, forming aggregates that are known to also recruit other proteins of the extracellular matrix. This can cause gain of function, loss of function, altering several pathways. And overall, it is suggested to trigger smooth muscle cell degeneration eventually. This will affect the whole vessel homeostasis and also causing dysfunction of the vessel. So the clinical consequences are ischemic strokes and cognitive decline, leading to dementia. Mutations are, the aggregates are systemic, but the clinical consequences are mostly in the brain. Considering that we need heterocellular interaction with endothelial cells, smooth muscle cells to activate the Notch3 pathway, also extracellular matrix plays a role, and this will affect eventually the whole vessel architecture. We don't need experimental models that recapitulate as much as possible all these different events. So in terms of the experimental models that are available at the moment, all of them provided very useful information, especially the transgenic mice, which we learn a lot, but we also know that incompletely reproduces the human phenotype. And when we want to work with patient-related material that is more related to the human situation itself, we do have also some useful information coming from primary brain vascular cells from CADASIL patients. However, those cells are limited source. They are difficult to expand as well, and also they need to be compared together with healthy control donors, and therefore here genetic background is not taken into consideration. Moreover, in terms of experimental approaches, most of the experiments are performed in 2D cell culture, which limits also this heterocellular interaction, the extracellular matrix formation, and therefore we do not having um, a situation that is close to the human blood vessel. And finally, human-induced pluripotent stem cells also came as an alternative approach for CADASIL. There are some studies, those are important as well, but most cases those studies compare data together with the healthy controls and not with isogenics. So again, genetic background here is a strong limitation. And we know that differentiated cells from induced pluripotent stem cells sometimes are often structurally and functionally mature, and they do require a more complex environment like tissue engineering in order to provide a better maturation of these cell types. So regarding pluripotent stem cells, they can be generated from somatic cells coming from patients. They are reprogrammed, and these are a powerful source to be differentiated to most of the cells of the body. And they have been used specifically for disease modeling, drug screening, and also toxicity tests. They provide several advantages in comparison to the primary cells. For instance, the availability to create many batches that can be cryopreserved, they are ready to go, and they are reproducible every time that we can generate them. Also, the pluripotent stem cells combined with organ-on-a-chip technology allows a tissue-specific environment that allows this recapitulation of conditions of the body, and specifically we'll focus on the vessel recapitulation. And finally, they are useful to generate new preclinical models, and this is the aim of this study, to allow us to study rare genetic diseases here in CADASIL in combination with organ-on-a-chip devices. So in this study, we want to combine pluripotent stem cell technology coming from CADASIL patients, then differentiate them towards vascular cells, endothelial cells, and vascular squamous cell cells, and integrate them into the organ-on-a-chip technology in order to recapitulate a 3D architecture. Here we see a representative image of endothelial cells, fluorescently labeled with M-cherry and small muscle cells in green. I will show you later how we can set up all of this. But this system allows this formation of a 3D structure that will promote the interaction between endothelial cells, small muscle cells, and activate the notch 3 pathway, which will be important to study CADASIL disease in the laboratory. So here it is the outlook that I would like to describe during this presentation. The final aim is, of course, the vessel-on-a-chip in combination with the pluripotent stem cells, but I will start with the generation of the lines, the CADASIL lines, generation of isogenic controls, differentiation towards vascular cells, and a deeper characterization of the iPS small muscle cells, and then generation of the vessel-on-a-chip, and finally another characterization of the small muscle cells now in the 3D setting. So regarding the pluripotent stem cell lines, they came from 2 CADASIL patients that carry the same mutation. It is associated with a severe phenotype and it affects the exon 4, a single point mutation. In total, I have been working with 2 clones of iPS coming from patient 1 and another clone coming from patient 2, and I use CRISPR-Cas technology in order to correct the mutation. So the mutation is a single point mutation in exon 4. It is heterozygous, and I use a guide RNA to recognize a region close by that will recruit the Cas9 protein, promote cleavage, and then use an SSODN that will be a template donor that will be used to repair the mutation and then generate an isogenic control. So overall, we're going to have 3 clones where the mutation is corrected, but still carries the same genetic background as the other middle lines. Then next step is to differentiate all these lines, isogenic and CADASIL, towards vascular derived lines by using protocols established in Valeria Orlova Lab. Regarding the small muscle cells have different developmental origins, and we're interested in the neurocast intermediates because small muscle cells that are in the vasculature in the brain, they do come from these neurocast intermediates. And for that, first we generate the neurocast cells, we expand them, and we have a pure population of passage 3. that is differentiated towards vascular smooth muscle cells by using TGF-beta3, which will promote the contractibility expression of genes, and the PDGFBP, which will promote the proliferation. And after 12 days, we're going to have a pure population that can be cryopreserved. Regarding endothelial cells, first it requires a mesoderm induction, followed by the addition of vascular endothelial growth factor and basic FGF to promote the endothelial cell lineage, and an isolation with the CD31 positive population, and having again a pure population and cryopreserved, which later on will be both of them incorporated into the microfluidic chip for the Persona chip. And importantly, we also perform bulk RNA sequencing from these 4 cell types and compared CADASIL versus control. And interestingly, we observed that the different cell types clustered well together independently. They were CADASIL or control. Also, they were different from other cell types. So we have IPS, neurocrest, smooth muscle cells, and endothelial cells. Regarding the neurocrest, we observed some differences in the cluster population, but they were more related from patient to patient one rather than CADASIL and control. And overall, we explored for differentially expressed genes, but they were pretty comparable between these 2 conditions. So overall, RNA sequencing confirmed that we successfully differentiated the different cell types. We generated IPS lines, CD31 for endothelial cells, Paxil for neurocrest, and calpain for smooth muscle cells, and there were no differences between control and CADASIL. So the next step that I perform is a deep exploration of the smooth muscle cells in 2D, exploring more genes from the RNA sequencing, which were pretty comparable. Those are contractile genes, SM22, calpain, or also the NEPS3 gene, which were pretty similar, except for actin cytoskeleton-related genes that there was some disbalance in CADASIL smooth muscle cells. And indeed, when we explore the smooth muscle cells by immunofluorescence, we can confirm that here SM22, again as a contractile marker, and NOTCH3, they seem pretty comparable between control and CADASIL. However, for F-actin cytoskeleton or other actin-related machinery like the calpain, there were some differences between the control and CADASIL. I could observe a more elongated distribution of the actin cytoskeleton, while it was more cortical related in the CADASIL, and also calpain seems to be more reduced. It's not the first time that it has been reported that there is some actin cytoskeleton dysregulation in CADASIL. There are some reports already from patients with muscle cells coming from the brain, so therefore it was interesting also to observe those changes in IPS smooth muscle cells. Importantly, also, I perform some functional characterization and explore how these smooth muscle cells will react. And for that, I measured the surface of smooth muscle cells changing into different situations upon mechanical pressure, that is basically moving refreshment, or by the addition of a vasoactive compound endothelin-1, which in both cases it resulted in the reduction of the self-surface of the smooth muscle cells, but this reduction was significantly lower in CADASIL smooth muscle cells, and therefore indicating that they were less responsive and less contractile. And also by measuring calcium dynamics by using a fluorochrome that will measure the calcium release over time, I could observe that there was some a slower response in the CADASIL smooth muscle cells upon applying mechanical pressure, while the response of calcium dynamics was pretty comparable and endothelin-1 vasoactive compound. So this 2D characterization showed that there were no major different gene expression or protein changes for contractile markers of NOTCH3. However, we observed some abnormal cytoskeleton structures and also some reduced contractility capacity and altered calcium dynamics. However, these smooth muscle cells still has not been exposed to heterocellular contact and into a more complex environment. And for that, I'm going to show you how Persona chip is going to play a role to promote the maturation of the smooth muscle cells, activate this pathway, and see what will happen with the cells here in this setting. So first of all, I would like to describe Persona chip technology by using iPS cells coming from healthy donors. It's a protocol that we recently described in stem cell reports, but also we showed that is a useful platform to study disease modeling as well by studying also a rare genetic vascular disease. The protocol requires the mixing of endothelial cells together with vascular muscle cells, 5 times higher concentration for endothelial cells, with fibrin hydrogel and incorporating into a microfluidic chip. The microfluidic chip that we are using, it is commercially available. It is the Hembiotech chip, and it is made of plastic. Each chip contains 3 different microfluidic channels, and in each microfluidic channel we can observe 3 different channels. The hydrogel channel, where we are incorporating the cell mix, and it is flanked by these 2 media channels. Then media refreshment can be added into these different ports, generating a gradient that will equilibrate over time, and this gradient of media will generate an interstitial flow that is important to promote the structures that we will see later. So now I'm going to show a time-lapse video. Here we do see a whole microfluidic channel, fluorescently labeled endothelial cells and smooth muscle cells in green. And we're going to see the cells self-distributing. The endothelial cells start forming vacuole formations that they will become more and more interconnected, forming these complex microvascular structures. And on the other hand, the smooth muscle cells that are in green, they will establish position, they will stabilize this structure, and they will start proliferating later on, stabilizing more and more all these network structures. So as I said before, we perform a daily refreshment of the microfluidic chip. We add the media supplemented with vascular endothelial growth factor, and also we add the gamma secretase inhibitor DAP that they want. That is known that in combination with the VGF to promote the formation of these sproutings. We do see most of the changes from these network structures from day 0 until day 5. And after that, it's mostly vessel stabilization, but also small muscle cells will start proliferating, assuming positions, and also stabilizing more this network. We stopped the experiments at day 7, but we know that we can extend this whole culture up to 21 days, and we believe also it can be spent even longer. So here I'm going to show you a video also from a region of interest made with confocal, again, these fluorescently labeled endothelial cells, surrounded by fluorescently labeled small muscle cells in green with the actin cytoskeleton. With a 3D reconstruction, we can properly see how this looks like, where we do see a nice interaction between these 2 cell types. The small muscle cells assume in position with the network. The vessels are also luminized, which is very important. We do see a nice barrier formation. The barrier is also confirmed to be stable as well. We confirm the integrity by perfusing dextran and beads. And overall, we see a nice harmony, a nice interaction between these 2 cell types. So in the same publication, we also show some properties on how to evaluate the characteristics of the vessel on the chip, and this will be important later on to show you how we can quantify changes in a disease setting. And here I'm showing again a whole microfluidic channel stained with glutenin, that is a marker from endothelial cells, and SN22, the contactile marker of small muscle cells. And we can segment these different cell types or properties by using specific markers. Vessel properties, I'm going to be using C31, bicadirin, SOC17 for nuclei of endothelial cells, and SM22 for small muscle cells. Then we use open source software in order to distinguish these structures and obtain parameters that later we use for analysis. We show as well that these vessels are luminized. Also, there is a nice interaction between these 2 cell types, as we can see here with the small muscle cells, but also there is extracellular matrix deposition in this model, and the vessels are perfusable, as we can show here, perfusing dextran, but also beads as well. So more importantly, also, we confirm that the small muscle cells in this vessel-on-a-chip setting, they do evolve, they mature, and they activate NOT3. And for that, we can observe here that the small muscle cells expressing this SM22 contactile marker, the ones that are in close proximity to the vessel network, do display a change of morphology and more intensity of this marker. And by using IMARIS technology, we can classify and obtain information of these different cell types. Here I'm showing in yellow the small muscle cells that are establishing contact with the microvascular network, and in parallel in magenta the ones that are not. And already we can see some morphological differences indicating that the small muscle cells that are in contact, they are maturing, they are changing morphology, and also they're expressing more SMT2. So SMT2 intensity was significantly increased in these small muscle cells. And also I explored how Notch3 would be changing in this setting. And interestingly, these cells that were also maturing more also displayed more Notch3, and it was correlating very well with this marker, and it was significantly increased, indicating therefore that there is a cellular communication leads to an activation and maturation of the smooth muscle cells, and also seems to be leading to a more expression of Notch3 and SM22. So now let's move forward to CADASIL differentiated cells. Again, we have these three CADASIL clones and the isogenic controls differentiated towards small muscle cells, so 6 lines in total, and in combination with endothelial cells using the same protocols I showed you before, and then generating the microfluidic chips. In total, I'm going to be showing data from 6 microfluidic chips performed by using different differentiation batches. But I'm going to be pulling the data in control versus CADASIL, but I will be displaying data from which clone is what in order to know if the data is clone dependent or it is really an effect from the NOT3 mutation. So first of all, evaluating the microvascular network parameters, where we can see here the network strain by CA31, comparing control versus CADASIL, and by eye we do not see major differences. They look very comparable. And also by evaluating some parameters like the vessel density, the diameter of the vessels, also the length, and also the amount of branches, we can observe that there are no differences between control and versus CADASIL, and also all the clones seems to behave in a similar way. So we do not see an influence from mutant smooth muscle cells directly into the microvascular network structure. However, when we pay close attention to the vascular muscle cells, first in the control we do see a more triangular shape, and also they are smaller, but in CADASIL, the vascular chip reveal profound changes, at least morphological in the smooth muscle cells. We're going to see now another 3D reconstruction to properly evaluate these changes. And we do see that the small muscle cells display these more elongated protrusions. They look kind of an hypertrophic situation. Some of the protrusions are also pointing out outside the vessels. The elongation is bigger, they look bigger as well. Also, it seems to be a higher proliferation as well. Vessels are luminized and seems to be stable. However, the small muscle cells seems to be more activated in this setting. So indeed, when I measured some morphological parameters like the length and the volume, they were significantly increased in the CADASIL small muscle cells, and this was observable in all the cultures that were incorporated in all the 3 different mutant clones. Next, I wanted to evaluate how these small muscle cells were changing in terms of phenotype. So some parameters that we explored, again, the SM22 contact marker, the Notch3, which is important here. And interestingly, the small muscle cells that are changing in terms of morphology, they seem to display higher SM22, higher Notch3 levels in comparison with the control. And when measuring the intensity, we do see indeed that the SM22 was significantly higher, and it was quite pronounced in comparison to the control, and also the Notch3. And this was also translated to the different clones, and it was observed and reproducible in the 2 patients. Moreover, these results were also correlated between Notch3 and SM22, and therefore indicating that when we do see this Notch3 accumulation of higher intensity, this also leads to morphological and changes and increment of the contact marker. Moreover, as I mentioned you before, there seems to be a high increase of proliferation of the small muscle cells, and indeed I found a significant increment of the amount of small muscle cells in CADASIL, And I mentioned you before that PDGF receptor beta, which is important for proliferation, it is also a downstream target from the NOTCH3. So we explored how PDGF receptor beta will look like in the vessel chip. And we also observed a significant increment of this marker the small muscle cells in comparison to the control, indicating as well that there is important changes of the phenotype of those cells. So the first results of this 3D vascular chip is that we will see some changes that were not revealed in the 2D setting, like morphological alterations, increase of number, and also increase SM22, NOTCH3, and also PDGF receptor beta. The small muscle cells in this setting seem to be changing phenotype and mature in a different way. And therefore, we wanted to explore a bit more the functionality of these cells and understand a bit more how they will behave in a visual chip. So we have previously shown that we can evaluate the small muscle cell functionality in the visual chip, and for that we focus on the cellular calcium release. And in order to evaluate that, we can engineer iPS small muscle cells that will express a forcibly labeled calmodulin protein that will bind the calcium and will release a green signal. Here I'm going to show a representative video of the small muscle cells forcibly labeled, and they're going to be perfused with media supplemented with vitamin 1. We do see a nice synchronized wave of the small muscle cells upon this exposure with media with vitamin 1 vasoactive compound. And by measuring this cell by cell, over time, we could confirm that small muscle cells exposed with vitamin 1, they were responding significantly high in comparison to the media, and therefore indicating that they are functional still, that they can respond to vasoactive components, and also that they preserve this contactile phenotype still. So next step was to evaluate this in CADASIL small muscle cells, and for that I focused only on patient 2, one mutant clone and one isogenic clone, again engineering small muscle cells that will express the same calcium sensor. First, I evaluated how these small muscle cells will behave in 2D, and for that I placed them into another chip. It is 2D culture, so we are having a monolayer of small muscle cells exposed by flow of endothelin 1, where we do see that in both cases, small muscle cells respond to this exposure of endothelin 1 and flow. Interestingly, however, the control, while it displays this main peak and some secondary peaks, the small muscle cells from CADASIL seems to be showing some disturbances and more pronounced secondary peaks as well. Next, I incorporated these small muscle cells into the microfluidic chip and perform at day 7 functional assays to evaluate how they will respond to flow and measuring over time this response. Here again, I'm going to show another representative video. We have controlled small muscle cells, CADASIL small muscle cells, and upon this perfusion, we're going to see that while the control show this main peak and they restore quickly to the baseline, the CADASIL small muscle cells seems to be fluctuating a lot, and they have this pronounced response with higher intensity. So I measured this for 2 experiments for now, so it's still some preliminary data, but I could observe that indeed in the control situation there is like a main peak that it recovers faster, while in CADASIL small muscle cells there seems to be a slower response, which resulted in significant increment amplitude over time, and also at time of 300 seconds it seems to be significantly higher. So it seems that the calcium dynamics of these small muscle cells are different in comparison with the control, and therefore the functionality and how they will regulate the vessel tone is also different. And finally, we also wanted to show that this model is a useful platform to study drug development, and we focus on the gamma secretase inhibitor tab, and we use it from day 5 until day 7. The idea here is to block gamma secretase, block the cleavage of the NOTCH3, and block the activation of the NOTCH3 pathway in general, and see how this will affect the small muscle cells in CADASIL. And indeed, we do see that the small muscle cells that are treated with the DAPT they do display less of these pronounced protrusions, also less than 22, and notch 3 seems to be less accumulated as well. By measuring some parameters like the volume and also the intensity of these two markers, I could find that it was significantly decreased and it was comparable to the control levels as well. Overall here, DAPT is of course a drug that will have a broad effect, so it cannot be used into the clinics, but we can show that the small muscle cells can respond to some stimulus, and therefore we hope that this platform can be a useful technology to study other drug developments. So the final conclusions in the 3D part is that this reveals profound changes in the small muscle cells. We observe morphological, protein levels, also in terms of functionality, and also that by applying the gamma secretase inhibitor DAPT, we can rescue some of these changes, and therefore this can be useful in the future. So regarding future perspectives, in the CADASIL research group, we previously shown that not only that notch variant position, it has an effect in the notch 3 aggregation, but also in the CADASIL disease severity. So we aim to show that the chip technology, it can be also a useful platform to understand what is the disease severity effect, and also in the future apply as well drug we're proposing to prevent disease development. So finally, I would like to thank, starting with cureCADASIL, also for awarding us with the grant last year, Netherlands of Organchip Initiative, Zonnebay in the Netherlands, and also CADASIL Research Group, Saskia, Julie, and the rest of the team, the Orlova team, Valeria Orlova, and also Theano specifically, who helped me a lot in the last 3D results. The anatomy and embryology department in general, but also specifically Christine Mamery, Ruben for the IPS RNA sequencing as well, and the IPS core facility who generated the lines and also characterized them. And finally, all of you for your attention. Thank you very much.
So thanks, Mark. This is great. I mean, I'm a clinician, okay, so I sit here looking at this and watching your presentation, and it's just you know, it's breathtaking. It's really cool, particularly if you know, as you've been pointing out, it can be a platform for looking at repurposed drugs or new drugs, or give us directions of where to go with this clinically. I don't see a lot of questions in the chat box, but I do have a couple of other things. One is this a way also to look at antibody to Notch 3? I mean, is that a possibility either before or after cell isolation? If you guys must have thought of that, or
as a to prevent notch accumulation, you mean, or
right?
It is one of the possibilities that we would like to test indeed. At this moment, we do not see like a formation of aggregates. So if the antibody is designed to target the aggregates, right now we will need to optimize this a bit more. But if we want to test at least how, in terms of notch 3 activation in general, cleavage, I think it can be a useful platform to see how the antibodies behave here. And at the end, it's the advantage of this is that you can also generate multiple chips coming from different donors, so you can validate this data as well. It's not coming from one specific result, so it's that's also the important part of using this platform.
Yeah, that's really cool. Were there any other groups that you know of that have done this or a similar model in CADASIL, or you guys are unique?
In terms of, there are some publications with pluripotent stem cells, so that's already nice. They do not incorporate yet the 3D model. They do more like a 2D exploration, but also they provide very valuable information. They study maybe like secreted factors from endothelial cells or small muscle cells, how they do affect each other. So they try to investigate some cross communication, but this really close-to-close interaction and changes over time. It's more new. But we used in the past already this last year we published that this model can be also used for study other rare genetic diseases. We do that with hemorrhagic telangiectasia as well.
Well, are there any other questions? Otherwise, I'm going to change over to my screen. So first off, I want to give an advertisement, okay, for an upcoming meeting. We, cureCADASIL, we're sponsoring a meeting at the United Leukodystrophy Foundation annual family conference. And it's going to be held in the morning for us here in the United States on June 24th. We've invited a number of investigators from the United States, you know, with NIH funding and whatnot, to come and do presentations and have an interaction with patients. So it's really a patient-driven meeting in which we'll have hopefully back and forth and interaction discussion with investigators. And I know that you guys are in Leiden, but it will also be virtual. You know, so if you go to our cureCADASIL.org website, if you're interested, you can register, and it will be virtual, so you can listen in on it. I'm not sure yet if they will have recording capabilities, but if so, we'll try to get it recorded and put it on our website. So I want to thank Mark and everyone else. We've had a small but great interested audience, I'm sure. We are, of course, going to in a day or two, this will appear on our website, and people will be able to access the recording and watch it. And usually we have quite a few people do that with these webinars. Please be sure to visit us online. And we are planning or thinking about having another webinar probably in July. And we'll, of course, post that to all of our mailing lists. And so once again, thank you, everyone. And without further ado, we'll sign off till the next webinar.